Hi everyone. In this video, I'm going to be talking to you about reabsorption in the loop of Henle. So up until this point, we've done ultrafiltration in the Bowman's capsule and the glomerulus, and then we did selective reabsorption in the PCT. And we talked about how 85% of all the water reabsorption occurs in the PCT, 100% of the glucose and amino acid and hormone reabsorption also occurs in the PCT, and most of our urea reabsorption occurs in the PCT. So now, let's just think about what happens in the loop of Henle. The loop of Henle is all about reducing volume of filtrate, reabsorbing water, and reabsorbing salts, sodium ions, chloride ions. So let's have a look. So here we can see that we've got the Bowman's capsule and then the PCT, and then we're leading down in the loop of Henle, descending limb of the loop of Henle, apex of the loop of Henle, and the ascending limb of the loop of Henle. So the walls of the descending limb are thinner than the walls of the ascending limb, and they are highly permeable to water, whereas the walls of the ascending limb are thicker and they are impermeable to water. So understanding this is essential to understanding how the loop of Henle works. The other thing that we really need to understand is sort of contextually where it is. So we know, for instance, that the whole of the loop of Henle and all the other parts of the nephron are completely surrounded by blood capillaries, but we can't just have these on the diagram because they would just get in the way and we wouldn't be able to see anything else that was going on. So we just have to accept that they're there. Okay, so you've got these blood capillaries surrounding the whole of the loop of Henle. In addition, there are other cells present, so there are other nephron cells. So all surrounding this area, we have got interstitial fluid. And we have got capillaries nearby. So in order to understand this, we're actually going to start in the ascending limb of the loop of Henle, not the descending limb. So in the ascending limb, sodium and chloride ions are pumped out of the nephron into the surrounding tissue fluid by active transport. Remember, the ascending limb is impermeable to water, so it cannot follow by osmosis. So in the ascending limb, the only thing that's happening, let's just have a quick look at it, is the sodium and chloride ions are being pumped out by active transport. This creates a salty solution around the nephron. This means the whole nephron is now bathed in a very salty solution with a very low water potential. This means that as the glomerular filtrate travels down the descending limb, water will move by osmosis down its water potential gradient across the wall of the descending limb of the loop of Henle, because it's permeable to water, and into the interstitial fluid. So as the solution travels down the descending limb, it loses water, and therefore it becomes more and more concentrated. Can you see these numbers here? Okay, so at first it's not very concentrated, but it becomes more and more concentrated as it travels down the descending limb because it is losing water. And why is it losing water? It's losing water because the solution is salty. And why is the solution salty? Why does it have a low water potential? Because we were actively transporting out sodium and chloride ions on the ascending limb side. So what's happening on the ascending limb? What's happening to the solution traveling in this direction is affecting what's happening to the solution traveling in this direction. This process, whenever you've got two solutions traveling in opposite directions and what's happening in one side is affecting what's happening in the other, i.e. the active transport of the sodium ions and the chloride ions, is increasing the movement of the water out of the nephron by osmosis. This is called a counter-current multiplier. You've come across this word before. So for instance, you learned about countercurrent multipliers in fish gills when the water and the blood travel in opposite directions, and that increases the absorption of oxygen into the bloodstream. So let's just think about where we are at now. So we've talked about the fact that as we move down the medulla, the solution gets more and more concentrated. That means a couple of things, right? One, on this side, we have got a very concentrated salty solution. And so here you have some passive transport of sodium and chloride ions out of the ascending limb. Almost all of the movement of sodium and chloride ions out is active transport, but there is a little bit of passive transport as well, just at the base. In addition, at the top of the descending limb, we've got a very watery solution. And so you've got some movement of sodium and chloride ions into the descending limb at the very top, down their concentration gradient by facilitated diffusion. So in the descending limb, there's two things that we're interested in: the osmosis of water out and a little tiny bit of facilitated diffusion of sodium and chloride ions in. And on the ascending limb, we are interested in a little tiny bit of passive transport of our sodium and chloride ions, and mostly the active transport of our sodium and chloride ions out of the ascending limb. So as we travel down the descending limb, we lose water. Therefore, traveling down the descending limb, we decrease the volume of our filtrate. And we increase the concentration of our filtrate. And then as we travel up the ascending limb, the volume doesn't really change because we're not really losing any water, but the concentration drops. So exactly the same over here. Can you see how here we're very concentrated, and with the same concentration here and here, you have the same concentration of solution leaving the loop of Henle as you had entering. You've just dropped the volume. So as you travel down the descending limb, you increase the concentration and decrease the volume of the solution. And as you travel up the ascending limb, you decrease the concentration and the volume remains about the same. If the same thing happens every time. So this isn't really osmoregulation, because you draw back in the same amount of water, you draw back in the same concentration of ions every single time. The only thing that can change this is the length of the loop. So some mammals have longer loops of Henle than others, and the longer the loop of Henle, the more space there is for reabsorption of water, and therefore the more concentrated and lower volume the urine that's produced will be. So longer loops mean more concentrated solutions, and shorter loops mean less concentrated solutions, because you'll have less time to reabsorb water. So I just wanted to end here with this diagram, because this sort of just closes it all off, shows you the whole process that is happening. What I'd like you to do is go through it, check you understand what's happening in the ascending limb and what's happening in the descending limb, how the volume and the concentration of the solution is changing at each stage, and how the length of the loop of Henle affects the urine that's produced at the end. But well done for listening to that, and I will speak to you soon.